question number 10, and it is a numerical answer type question which carries 1 marks and it has no negative marking. Therefore, it is always advisable to answer this type of question. So the question here is that 1 mole of a circular plasmid DNA. So there are certain things you have to know, that there is 1 mole, the starting material is 1 mole, and it is a circular bacterial plasmid DNA. It was digested with the high fidelity restriction enzyme. So it is a high fidelity restriction enzyme. The plasmid has 5 restriction sites for the enzyme routes. So this thing also 5 restriction sites, and you have to calculate the number of moles of fragments released upon cleavage at all the sites. So to answer this question, you first have to know the basic about the restriction enzyme. So if say suppose you take the restriction enzyme ECO R1. So what is the basic principle behind a restriction enzyme is that it recognizes a particular sequence of DNA. That is, like this ECO R1 restriction enzyme will recognize whenever there is a GAATTC site in the DNA. So then it will recognize the site and it will cleave after the G residue in both the strands. So and then after that it will create something like this, that is both the strands are cleaved after the G. Here this side and this side, and it will produce a sticky end. Right, this 5 prime end is overhanging, and its length is longer than the 3 prime hydroxyl end. So restriction enzyme cuts the sugar phosphate backbones and creates sticky end or cleaved DNA fragment. But this is the sticky end produced by restriction enzyme ECO R1. But there are different kinds of restriction enzymes, and they produce different kinds of ends. Says like for example, ECO R5, it recognizes this DNA sequence and it cleaves after T, so it produces a restriction fragments which are equal in length, both the 5 prime and 3 prime end, and therefore it produces a blunt ended fragment. Then we have ECO R1, which I just explained, and then we have PSD1, which produces a 3 prime overhang, that is the 3 prime hydroxyl group side is longer than the 5 prime. Then we come to the next part of the question, that is, we have a circular plasmid DNA, which is mentioned in the question. And there are 5 restriction sites for that enzyme. That is, so it will cleave at, and because it is a high fidelity restriction enzyme, so it will cleave at all the 5 locations with equal affinity and equal efficiency. So after cleavage at all the 5 sites, the number of fragments we will receive, if there is one circular plasmid, the number of fragments received will be 1, 2, 3, 4 and 5. So if the starting material is 1, then we will get 5 fragments, and here in the question the starting material is 1 mole. So if we start with 1 mole, then we will receive 5 moles of the fragment upon cleavage at all sites. So this is about the circular plasmid DNA. But if, for example, if we get a question in which the circular is replaced by the linear DNA fragment, say suppose a PCR product, and everything else is same. So the restriction sites are 5 and the enzyme is high fidelity. So there the number of fragments will be not 5, but it will be 1, 2, 3, 4, 5 and 6. So if the starting material was 1 mole, then you will get 6 moles if it is a linear DNA fragment. But if it is a circular DNA fragment, then you will receive the equal number of moles as there are number of moles of fragment as their number of cleavage sites. And another important thing is that it has to be a high fidelity restriction enzyme which cleaves at all the site. Then only these two conditions, if these two conditions are fulfilled, then only you will get equal number of fragments as there are equal as many restriction sites are present in that DNA fragment.